For degenerative diseases such as cardiomyopathy, regenerative therapy using induced pluripotent stem cell-derived cells has massive potential. However, in vitro accelerated cell growth leads to DNA damage by accumulated metabolites such as reactive oxygen species. Transplanting these DNA damaged cells would result in poor engraftment and regeneration of the organ. Before transplantation, the quality of the cells should be assessed. Here, we provide two step-by-step protocols to assess the DNA damage in stem cells. Demonstrating the procedures will be Jessica Miller, a researcher, Nikhil Mardhekar, a researcher, and Vasanthi Rajasekaran, a lab technician.
To begin, place 500 milligrams of low-melting agarose in 100 milliliters of DNA-RNA-free water. Heat the bottle in a microwave oven until the agarose dissolves. Then, place the bottle in a 37 degrees Celsius water bath until needed. To prepare pre-coated comet slides, place a few drops of 0.5% agarose on a glass slide. Immediately spread the agarose to form a thin layer of agarose coating using a cover slip. Working under low-light conditions to avoid ultraviolet light-induced cell damage, mix 10 microliters of cell suspension and 90 microliters of agarose solution in a tube. Place the tube in a 37 degrees Celsius water bath to prevent the agarose from solidifying. Mix the solution without introducing air bubbles and pipette 70 microliters per spot onto the pre-coated comet slide. Using a pipette tip, spread the cell agarose mixture to form a thin layer. Place the slide at 4 degrees Celsius for 15 minutes.